Hi there, my name is Courtney Richardson, and I'm going to be doing a presentation today. So let's get started with sharing the screen. And okay, so the presentation today is on glycolysis, gluconeogenesis, and the role of aldolase in gene expression. Excuse me, I have allergies. So let's get started. Okay, so glycolysis is a cytoplasmic pathway that breaks down glucose into 2 3-carbon compounds and creates energy. It takes place in 10 steps, which are broken down into 5 preparatory steps and 5 payoff steps. Glycolysis is used by all of the cells in the body for energy production. The final product is pyruvate in aerobic conditions and lactate in anaerobic conditions. So the first step of the glycolysis process, you can see right here, is the conversion of D-glucose, which is the most commonly occurring natural simple sugar, then into glucose 6-phosphate. So recall that the 6 is the position, the phosphorylation of glucose. It occurs on that 6th carbon in our carbon ring, as you can see here. Phosphorylation is the process of adding that phosphate group to a molecule derived from an ATP. So 1 molecule of ATP has been used up in this step and is now converted to ADP. The enzyme hectokinase is used in this first step, and it catalyzes the phosphorylation of the glucose ring. This first step is glucose 9-phosphate with the 6th carbon of the glucose acquiring the phosphate group taken. I'm sorry, 6-phosphate, which has this 6th carbon on the ring acquiring that phosphate group. So let's move on to our second step. The second step is the conversion of glucose 6-phosphate to fructose 6-phosphate with the enzyme phosphoglucose isomerase. So remember that the word isomerase implies that the process of isomerization, which is a term that basically means that the molecule is just kind of reconfigured to a different physical structure, although it kind of has the same chemical makeup. The isomerization reaction occurs when the 6th carbon ring opens and then shifts to close one of the carbon molecules outside the ring. So we can see that here. It shifts, and now we have 5 carbons on this ring instead of 6, giving it the fructose name. And also we have that carbon right here, as you can see, so fructose 6-phosphate right there. So now let's move into the third step, which you can see right here. Let me move my little camera. Oh no, you can see that. Okay. The third step in the process involves the enzyme phosphofructokinase. It's kind of blurry, but you can see that's phosphofructokinase right there. So phosphofructokinase with magnesium as a cofactor actually is what we're going to be using in this step. So remember, cofactors are chemical compounds or ions that are required for an enzyme to work as that catalyst. So this step here works kind of similarly to the first step of glycolysis in that the second molecule of ATP donates its phosphate group. Here we have again ATP, it goes to ADP, donating that phosphate group, which is then added on to the first position here, which is that, if you recall in the nomenclature, that 1 is distinguished here because this is our first carbon that's added that phosphate group. So that's why it is fructose. How many? 1, 2, 3, 4, 5. So that's our fructose. And then 1 is our first phosphate group that's been added, and then 6 in the 6th position biphosphate. So, so that is going to be step 3. Okay. And then let's move on. Perfect. So let's move on to our next slide. Here we go. So I've uncovered a little bit more, and I'm going to move myself right there so that way you can see this part a little bit better. So now we have steps 4, 5, and 6. We're just going to start with 4 right now. So step 4 has the enzyme Aldolase. You'll see right here, which splits fructose 1,6 biphosphate into 2 sugars that are isomers of each other. Aldolase catalyzes the cleavage of fructose 1,6 biphosphate to yield 2 3-carbon molecules. One is called glyceraldehyde 3-phosphate, and the other is dihydroxyacetone phosphate. Moving on to step 5, the enzyme triose isomerase rapidly interconverts with the 2 isomer sugars dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate is the only molecule that continues with the glycolytic pathway. All of the other dihydroxyacetone phosphates molecules are then changed into glyceraldehyde 3-phosphates by the enzyme triophosphate isomerase. This reaction ensures that all of the products that are used so far in the glycolytic pathway are actually going to be productive instead of wasted. So now we have 3 carbon molecules, and we're going to move on to step 6. And in step 6, it kind of can be broken down into 2 little mini steps. So we're going to go over those now. So the first part of it is the glyceraldehyde 3-phosphate is oxidized by the coenzyme nicotinamide. I'm sorry, I read that incorrectly. Nicotinamide adenine dinucleotide. And then the molecule is then phosphorylated by adding a free phosphate group. This is all catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. This enzyme also positions the molecule in a way that allows for the nicotinamide adenine dinucleotide molecule to take a hydrogen from the glyceraldehyde 3-phosphate, turning it into nicotinamide, I cannot say that word, I'm so sorry, adenine dinucleotide plus a hydrogen, also known as NADH, which is a little bit easier for me to pronounce right now. So the glyceraldehyde 3-phosphate is then released from the enzyme by the phosphate group, which yields 1,3-biphosphoglysarate, a hydrogen atom, and the nicotinamide adenine dinucleotide with the hydrogen, NADH. So you can see that right here. So let's move on to our next slide, and I'm going to move myself up here into the corner because we already covered this section. So step 7 begins with the 1,3-biphosphoglysarate being converted into 3-phosphoglycerate by the enzyme phosphoglycerate kinase. This consists of the loss of the phosphate group off the 1,3-biphosphoglycerate. This phosphate is then transferred into a molecule of ADP, creating the first ATP molecule. And then this actually occurs twice because if we remember, the 2 3-carbon products at the beginning of the process is actually 2. So because the process of glycolysis has already invested 2 molecules of ATP, we have now zeroed out our investment in this stage back to 0. So step 8, and this is why it is called the payoff phase, because step 8 involves the rearrangement of the position of the phosphate group on the 3-phosphoglycerate molecule, creating 2-phosphoglycerate. This reaction is catalyzed by the enzyme phosphoglycerate mutase. So if we remember, mutase is an enzyme which catalyzes the transfer of a functional group from one area to another on that molecule without changing the chemical. This occurs by placing an additional phosphate group on the 3-phosphoglycerate on the 2 position, the second carbon position. So the enzyme then removes the phosphate from the 3 position on the 3rd carbon and only the phosphate in the 2 position, which leaves it as 2-phosphoglycerate. The enzyme is also restored to its original state during this process. Step 9 involves the conversion of the 2-phosphoglycerate to phosphoenolpyruvate, which is catalyzed in the enzyme enolase. Enolase dehydrates the 2-phosphoglycerate by removing a water group, and the reaction occurs through a series of complex steps due to the specificity of the enzyme pocket. So the final step is step 10, and this converts the phosphoenolpyruvate with the help of the enzyme pyruvate kinase. This reaction involves the transfer of a phosphate group, which is attached to the 2-position carbon of the phosphoenolpyruvate, which is then transferred to a molecule of ADP, creating another molecule of ATP. If we recall, this reaction is happening with 2 molecules of phosphoenolpyruvate. So there's actually, again, 2 molecules of ATP generated. So this is a little bit of an easier way to kind of look at it. We have the enzymes on this side, we have our carbon structure on this side, and our names. The cell continues respiration at this point, either anaerobically without oxygen or aerobically, which is with the presence of oxygen. If the cell does continue with aerobic respiration, the final output is going to be, it continues to the citric acid cycle, and then I think so it continues on to this way, and then anaerobic respiration then produces, it goes into fermentation, and then it produces ethanol and lactate. Okay, so let's move on to gluconeogenesis, and we're going to go over this now. So the term gluconeogenesis, let me move myself again, there we go. The term gluconeogenesis refers to a group of metabolic processes which are regulated by insulin, cytosol, and the mitochondria. The purpose of gluconeogenesis is to maintain blood glucose levels in a constant state throughout periods of time when the body isn't getting food, also known as fasting periods. Many parts of the body require continuous glucose supply when the body isn't getting food, because you're not going to eat constantly. That's not realistic. So these parts of the body that require a constant supply of glucose are your skeletal muscles when you're moving, your brain, which is always taking in information and processing, and as well as your red blood cells. During the first few hours in a fasting period, I'm sorry, I lost my place. The glycolysis is the primary glucose source. When glucose is stored as glycogen, it can cover the basic glucose for about a day. However, after several hours without food, the gluconeogenesis and the glycogenolysis both equally contribute to blood sugars, to the blood's sugar supply, glucose supply. Once the glycogen stores deplete, the body then uses glucose as a source of lactate, glycerol, glucogenic amino acids, and fatty acids. Skeletal muscles produce aniline through protein catabolism, which is then transported out into the bloodstream and taken to the liver, where the aniline undergoes transamination into pyruvate, which is then used for the gluconeogenesis. Glucose is also produced in the liver and transported through the bloodstream when used by skeletal muscles in the ATP production. In red blood cells, which lack mitochondria, glucose is converted into pyruvate and then into lactate. Lactate is secreted into the plasma and then also taken to the liver for conversion into glucose. This occurs through a redox reaction, which is catalyzed by lactate dehydrogenase. dehydrogenase. Triglycerides are mobilized by lipase, and the glycerol from the triglyceride structure is then released into the circulatory system, also to be taken to the liver, and then phosphorylated by the glycerol kinase and oxidized into dihydroxyacetone phosphate. Dihydroxyacetone phosphate is an intermediate of the glyconeogenesis and glycolysis pathways by glycerol phosphate dehydrogenase. Off-chain fatty acids are converted with beta oxidation into the propanol CoA. Even chain fatty acids, along with purely ketogenic amino acids, are converted into acetyl-CoA, are unable to enter gluconeogenesis because there is not a pathway that can reverse the catalyzation by pyruvate dehydrogenase. So in review, glucagon regulates gluconeogenesis in 3 ways, through the changes in allosteric regulators, through covalent modification of enzyme activity, and through the induction of enzymes gene expression. Acetyl-CoA activates pyruvate carboxylase allosterically, and gluconeogenesis can be increased or decreased through substrate availability. For gluconeogenesis to occur, the ADP and ATP ratio must be low because gluconeogenesis being a highly demanding process, so it requires a lot of energy molecules to be spent because it in many of the steps that it undergoes. Okay, let's move on to Aldolase. And so Aldolase is a tetramer and can be found in all eukaryotic cells. It is essential for the metabolism in fructose and present in both the glycolysis and gluconeogenesis pathways. In glycolysis, Aldolase helps break down glucose into energy in the 4th step. It breaks down fructose 1,6-5-phosphate into dihydroxyacetone phosphate and glycerolide 3-phosphate. In gluconeogenesis, it helps to convert glycerolide 3-phosphate into fructose 1,6-5-phosphate. The expression of Aldolase can be divided into 3 isosomes, which are encoded by 3 genes that originate from a single ancestral gene. Aldolase A is a primary isozyme in mammalian embryonic tissues, for example, while Aldolase C is found to be the primary embryonic form in birds and amphibians. Aldolase B is mainly found in human livers, kidneys, and there's actually even a small amount in the intestinal mucosa. So there's our A, B, and C. So we're going to move on to Aldolase B now. So Aldolase B isozyme mutations can cause autosomal recessive diseases, hereditary fructose intolerance, which is very serious. Hereditary fructose intolerance can lead to excessive levels of fructose 1-phosphate in the tissues that express Aldolase B, leading to liver and kidney damage. The transcription of the Aldolase B gene is stimulated by insulin and glucose. CEBP is a protein which regulates cellular proliferation and differentiation through mechanisms. HNF1 and 3 are hepatocyte nuclear factor proteins and regulate the metabolism and differentiation of metabolic tissues. CBP is a protein coding gene. So now we are going to examine how these transcription factors regulate the expression of Aldolase B. So Aldolase B promoter is controlled by 5 elements from the 5 to the 3 end. In the process, there is an activating EBP-DBP binding site and an activating element which consists of 2 overlapping binding sites for HNF1 and HNF3. CEBP is also considered to be a weak activator in the HapG2 cells of Aldolase B. Additionally, the deletion of the distal negative element results in similar transactivation by CEBP and DBP. CEBP and DBP activation may also depend on the interaction of tissue-specific proteins binding to neighboring sites. HNF3 acts as a strong anti-activator of the promoter of Aldolase B, which then concludes our examination of how Aldolase B expression is regulated through transcription factors HNF1 and HNF3 and CEBP and DEP. So there are all the references in full. And so in the summary, we covered an overview of the glycolysis and gluconeogenesis pathways. We examined how Aldolase is involved in those pathways, and then we looked into the different isoforms of Aldolase and their expression patterns. We also examined how Aldolase B specifically expression is regulated by a few transcription factors. So thank you for listening to my presentation, and